Okay, so bacterial development. This might seem like an oxymoron to you. If you take developmental biology here at NC State, you're going to talk about chickens and stuff like that, right? Maybe even people. Flies. In other words, animals. Maybe you talk about plants. Bacteria also have development. What is development? Development is the transformation of a cell from one phenotype to another. Whether that's from a stem cell to a muscle fiber or whatever, that's development. It's a transformation of gene expression and usually associated with a transformation in the structure or function of the cell. Isn't the transformation of an E. coli from logarithmic growth to stationary growth a developmental process? Sure it is. Gene expression is completely transformed in that process. And it's completely transformed because in logarithmic growth, you have sigma 70 that's controlling the expression of all the genes. And then when you're starved, you turn off sigma 70 and turn on sigma 35, a completely different transcription factor. And it has an entirely different suite of genes that it turns on. The evolutionary pressures on logarithmic cells are very different than those on stationary cells. In logarithmic growth, your task is to get stuff out of the environment as fast as you can, usually it's abundant, and make cytoplasm and divide as fast as you can. Stationary phase cells have to typically extract trace nutrients out of the environment. They're not interested in division, often not interested in growth. They're interested in survival. And so they extract trace nutrients, they do what metabolism they need to, they keep their DNA repaired, but they're not growing, increasing mass or anything like that. Those are two very different situations. Phage infection is a stereotypical developmental process. Most phage have a series of sigma factors that they encode, and each one controls each stage of the viral infection process. You might have an infection sigma, an early expression sigma, a middle infection sigma, and a late infection sigma. Obviously, the early ones are involved in replication of the genome, and the late ones are involved in making all the proteins you need to encapsulate and be released from the host. This thing, streptomyces, that's the developmental process. Anabina, heterocyst formation in anabina, aconite formation in anabina. These are all developmental processes. Development is a fundamental aspect not just of eukaryotes, but of all creatures.